We humans have these extremely important cooperative relationships called friendships, which have a number of traits that are interesting to a behavioral ecologist. Our friendships are cooperative and individualized. We help each other, even if this takes time and energy. Now, there's always a conflict in social relationships, but friends also have long-term interests in each other's survival and well-being. There's interdependence. Interacting with friends creates positive emotions, which allows us to exchange multiple kinds of services or favors by integrating these rewarding experiences over time. Some other species also have social bonds, with some or all of these traits. We can ask about their adaptive function, the mechanisms that regulate them, and in how many lineages they evolved. But we know very little about how social bonds actually form between strangers. Animals often choose their social partners. Other times, they're simply born into a relationship. For example, a female juvenile bat starts off with one vitally important bond, with her mother. As a juvenile vampire bat grows up, she also begins forming bonds with non-kin. These bonds involve many of the same interactions she had with her mother, like clustering for warmth, reciprocal grooming, and food sharing. Food sharing involves a fed bat regurgitating a portion of her blood meal to help an unfed bat. But how do these bonds form between strangers? To find out, we've been collecting bats from distant sites and introducing them. In one experiment, we kept two unfamiliar groups separate, although they could vocally interact. Next, we introduced pairs in small cages, where we introduced one bat to three others in a small group. Finally, we threw everyone together. And in every phase of this experiment, we induced and observed grooming and food sharing. Over 13 months, the probability of new food sharing among two adult females was only 15%, but most new pairs began social grooming. Adult females gained on average 1 or 2 new food donors, whereas captive-born juveniles gained 2 or 3 new food donors. By the end, the average female bat had a total of 7 donors in her network. This plot shows the accumulation of first donations over time. Some relationships never formed. Others did not form for many months. These relationships often formed in a reciprocal fashion. If bat A fed B, it was more likely that bat B would later feed A. The same was true of grooming. As expected, we saw that new food-sharing bonds formed faster in isolated pairs and quartets. Now, these results were published earlier this year, and I've presented these results before, so I'll go through these plots quickly, but please just pause the video to take a longer look if you need. We saw that increasing grooming rates predicted new food sharing. In other words, a food donor had a history of being groomed increasingly by the bat she fed. This pattern only existed in pairs that formed sharing bonds, but not in pairs that did not. We saw that grooming rates increased up until the start of food sharing within each pair. Do these social experiences actually matter? Could it be that bonding is based merely on phenotypic similarity? In other words, do some pairs of bats just immediately, quote, bond because, for instance, they smell similar? Or does it actually take time? Can we predict future rates of grooming and sharing from how two strangers interacted when they first met? To answer this question, 2 undergrads in my lab, David and Jessica, went back to the raw video footage of when I first put the bats together. David and Jessica had no idea of the social future of these bats. Their job was to try and predict which ones would later share food from their first 6 hours together. They measured fighting, sniffing, grooming, and mouth licking, or begging. We then correlated these measures with the long-term outcomes which we already knew from the previous study. To test the hypothesis that first impressions might predict the future, we compared our observed effect sizes with those expected from a null model of what could have happened by chance. To generate these expected null effect sizes, we randomized the interaction rates of each introduced bat across her possible partners. In other words, we swapped the actual partner bats, their labels, with other potential partners, and then did the same analysis to get an expected effect size. We did this 5,000 times. The take-home message is that we could not easily predict long-term relationships from the first 6 hours of interactions. None of the measures taken by David or Jessica could clearly predict grooming or food sharing rates months later. This plot shows the effect of the overall affiliation, which includes sniffing, grooming, and begging. The observed value, shown in black, falls within the distribution expected by our null models, shown in gray. So what can we conclude? The initial interactions, or first impressions, probably do matter, but not that much. In this small sample of only 20 bats, the first 6 hours do not predict the future. Now, this does not tell us that the initial interactions don't have any information or that they don't tell us anything. It only tells us that their long-term social bonds were not entirely predictable from these first impressions. This is entirely consistent with the idea that social history actually matters. Next, we are now getting more relationship trajectory data from more bats, and we will be using proximity sensors to look at new relationship formation in a less structured group setting. In this setup, we should be able to get more reliable measures of effect sizes. Thank you very much.